I hope everybody's enjoyed the first 2 days of tips and this morning's sessions, all of which have been really excellent. And I think by and large have focused on the human applications of intensive and digital-based methods for assessment and interventions in psychiatry. But we're really excited today to really feature how some of these same approaches, both on the data acquisition side, but also especially on the computational modeling side, are applying in animal neuroscience as well. And so Bob is going to talk to us today about some of his work, which really tries to show how these same approaches can be applied not just to clinical applications, but really to improving the granularity and interpretability of neuroscience. And so Bob is a professor of neurobiology at Harvard Medical School. The goal of his laboratory is to address how the brain extracts sensory information from the environment and convert that information into actions. By studying and characterizing particularly the olfactory, sensory, and motor circuits in mice, the Datta laboratory aims to understand how neural codes for sensation and action are built and how they generate organized and goal-oriented behaviors, including both survival-based and social behaviors. I'm particularly interested in Bob's work as he studies animals in more wild settings, or at least in simulated wild settings, over long periods of time, which is something that's also near and dear to my heart as applied to humans. He's received many awards in his career, including the NIH New Innovator Award, the Burroughs Wellcome Career Award for Medical Scientists, and the Searle Scholars Award. And with that, please join me in welcoming Bob Datta. And Bob, please take it away.
Thank you, Justin, for that invitation. Are we good in terms of hearing me and seeing me?
Looks great.
Excellent. Well, thanks everyone for joining. It's a treat to be able to share some of my lab's work, especially in front of an audience that I don't usually get the opportunity to speak in front of. As Justin mentioned, I'm going to give you a basic sciencey talk, but I think there are substantial implications, both for the ways in which we build drugs for psychiatry and for our understanding potentially of psychiatric disorders themselves. And I'll highlight the few ways in which that's true during my talk. But it also means that I have to give you some disclosures because some of the technologies that I'm going to describe to you today are being kind of deployed in the drug development setting. And those include disclosures related to Neumora Inc., who's one of the sponsors of this meeting, and Gilgamesh Therapeutics. Okay, so let's get going. So when you watch animals behave in the natural world, it's difficult not to be struck by the beauty of their behavior. And also its complexity. It's often just by looking, it seems as if animal behavior is kind of inscrutable. We don't really understand what or why the animals are doing when they're free to do what they want, when they want. But the ethologists thought they could make sense of this kind of behavior, and they hypothesized that animal behavior is actually composed out of a series of behavioral modules, each one of which is a brief, three-dimensional, stereotyped, and reused motif of action that the animal can place into series to compose much more complicated patterns of action, which also are organized hierarchically in time. And this suggests a kind of underlying spatiotemporal structure that governs natural spontaneous behavior and which allows animals to meaningfully interact with their environment. But despite the fact that this sentence was now written nearly 50 years ago, I think it remains unclear how it is the brain actually, on a moment-by-moment basis, composes natural spontaneous behavior. And by that, I mean the kinds of behaviors that animals express when they're free of physical or psychological restraint. I'm talking about the kinds of behaviors that we as humans express in most of our lives, the kind of behaviors that happen without being put into an explicit task and being explicitly rewarded or explicitly punished. And so we've wondered for many years about how animals and humans create kind of spontaneous naturalistic patterns of behavior. And so we've been building machine vision and machine learning tools whose aim is to unveil the underlying modular architecture of mouse behavior. The tool we've built is called motion sequencing, or MoSeq for short. We've published extensively, as you can see on the lower right, on both how the technology works and some of the applications of the technology. So I'm not going to dwell on the kind of methodological details here. I'll just at a very high level give you a sense of how motion sequencing works. So MoSeq takes in as its input really any form of time series behavioral data. And then through unsupervised machine learning techniques, based on the structure and patterns of action that are expressed in the data alone, MoSeq automatically identifies the optimal set of behavioral modules, or what we refer to as behavioral syllables, out of which behavior is created in any given experiment. Now, you can feed almost any type of behavioral data into MoSeq, and we've done all sorts of things. We recently published a paper with my friend and colleague Lisa Stowers, actually analyzing mouse song using MoSeq. But typically, we feed MoSeq the kinds of data you're seeing here on the upper left. You're looking at 3D imaging of a mouse as it freely explores an empty circular open field. Basically, we throw a mouse in a bucket, and then we use depth cameras, which from a single axis of view you can see in 3D, to image the mouse's overall pose dynamics. And so heat mapped over each pixel of this mouse, this freely behaving mouse, is actually the height of the mouse. And so when the mouse's head turns red, as it just did, white, as it just did there, it means the mouse is rearing up. When the mouse's back turns white, it means that the mouse's back is arched up towards the camera. And we feed these data to MoSeq, which in this unsupervised way identifies a kind of parts list for behavior, a set of syllables that describes behavior. For each frame, MoSeq will identify which syllable is expressed. The number of syllables in a typical open field experiment varies depending on the specifics of the experiment, because this, again, is an unsupervised technique. So MoSeq will discover maybe 40, 50 of these behavioral syllables in a typical open field experiment. And each one lasts roughly 300 milliseconds, although there's quite a bit of diversity of how long these behavioral syllables last. And so for a given behavioral video, MoSeq will spit out these labels, as you can see here on the lower right, identifying specifically which behavioral syllables being expressed at any moment in time, as well as the order in which they occur. And that allows us to take all of this information and synthesize it into what you're seeing on the lower left, which is, of course, a behavioral state map. Here, each node represents a behavioral syllable that's been discovered by motion sequencing within an experiment. The diameter of that node reflects how often that syllable is used, and the thickness of these yellow directional arrows indicates the observed likelihood of transition over time between one syllable and the next. And so these behavioral state maps do a really wonderful job of quantitatively synthesizing and representing the complete behavioral deportment, the naturalistic behavioral deportment of an animal within any given experiment. And as such, we can use these quantitative holistic descriptions of behavior to ask how various experimental manipulations might have changed the animal's behavior. And we've done this in every setting you can imagine. We can give sensory cues to the animal or change the animal's environment and watch how its behavior changes. As I'll show you in a bit, we can use pharmacogenetics or optogenetics to tweak activity in different neural circuits. And of course, we can manipulate genes, as I'll also show you, and understand how our genetic manipulation has changed the overall architecture of behavior. One of the things that we've used this technology for, which I'll just briefly mention, is to better understand the influence of psychiatric drugs and neuropsychiatric drugs on behavior. And so you can give all of the drugs basically in the modern neuropsychiatric pharmacopoeia to mice, image them in 3D through motion sequencing, and then characterize their behavior and ask, in what ways are drugs that are thought to target the same mechanism different? And you can also ask about kind of global similarities in the behavioral effects of drugs. And so you can build drug maps like this. I won't get into the details of this map, except just to say that one of the really wonderful consequences of describing behavior as being built out of modules and sequences is that it very naturally captures not just the kind of target-based relationships amongst drugs, but the relationships amongst drugs that are actually observed by folks who are working in the clinic. And so many of the relationships here that you're observing, are amongst drugs that actually have very different mechanisms, but seem to converge upon similar phenotypes when applied clinically. We can also use this technology and especially its resolution to better understand how drugs might influence or ameliorate disease phenotypes in mouse models of disease. And I'll just give you one quick published example of this. Here we're going to phenotype the CatNap2 mouse model of autism spectrum disorder. So CatNap2 is a gene when mutated in humans is causal for autism. And it's known both in humans and in mice that mutation of the CADNAP2 gene confers upon those that harbor this mutation a kind of hyperactivity phenotype. What you're looking at here in the upper panel is called a usage plot. It's actually just a small subset of a usage plot. The x-axis, each tick represents an individual syllable that has been identified by motion sequencing, and the y-axis simply represents how often that syllable is used. And what you can see is there's a whole host of syllables that appear upregulated in their usage in the CADNAP mutant relative to just wild type animals. And when you inspect these syllables, they clearly represent different behavioral motifs that are underlying the well-documented hyperactivity phenotype that's exhibited by these mice. Now, based upon our drug database and based upon the specific syllables that we identified as being upregulated here in the CADNAP2 mutant, you can basically intersect these data sets and identify candidate drugs that might actually revert this mutant phenotype back to normal. And so we've done that with a number of drugs for the CADNAP2. I'm just going to show you one kind of illustrative example, which is this drug loxapine, which as far as I know hasn't been deployed clinically to treat ASDs, but based upon this kind of intersection, seems like it would be a reasonable choice. On the lower plot here, you're looking at the effects of loxapine on the behavior exhibited by CADNAP2 mice in yellow. Underneath each syllable, you can see either a little circle, an X, or a square. X's represent syllables that weren't reverted by treatment with loxapine. The circles represent those syllables that were completely rendered normal due to treatment with loxapine. And the square represents syllables that were previously normal in the mutant, but now are abnormal under loxapine. And so you can think about these as being missed effects, on-target effects, and actually side effects. And so you can see that by atomizing behavior through the lens of motion sequencing, we get this really fine-grained picture of what's going on behaviorally, and we can, I think, much better understand the effects of candidate therapeutics. And as I mentioned, this is something that's going on clinically at both of those companies that I'm involved in. So now I want to turn back for a moment to basic science and just describe to you that one of the key questions that my lab has been working on over the last several years. So this is one of these behavioral state maps for a C57 mouse, and it's clear that this map contains within it an enormous amount of structure. It's obvious the brain is articulating rules that govern how often each syllable is used and the order in which they occur. And a question that we've had now for many years is, from where does the structure arise? How is the brain actually regulating, how often syllables are used and the order in which they occur during any given experiment or context? And that naturally got us thinking about the basal ganglia, and in particular, the input nucleus to the basal ganglia, the striatum. And that's because, of course, the striatum is well known for its key role in regulating various aspects of movement, and as well, it has well-defined roles in a variety of settings in action selection, which is, of course, the process through which animals decide what to do next. And you might imagine that action selection, like mechanisms, could be important in selecting which syllable to express in a sequence. We've decided to focus specifically on the dorsolateral striatum, and that's for really two reasons. The first is that the dorsolateral striatum collects inputs from sensory motor cortices, and that really positions it to potentially encode useful information about an animal's ongoing three-dimensional pose dynamics, which is, of course, what these syllables are made of. But the other thing is that pioneering work from Kent Berridge and John Fentress and others in the 70s and 80s really suggested that the dorsolateral striatum might play a key role in encoding and implementing sequenced natural behaviors. And they focused on a particular behavior that's expressed by rodents, which is grooming behavior, which has been shown to be sequential and doesn't require training. And so we hypothesized that perhaps actually the dorsolateral striatum is a kind of general purpose machine for controlling 3D pose dynamics and their sequencing. And so in order to test that hypothesis, we wanted to do neural recordings of activity in spiny projection neurons within the dorsolateral striatum simultaneously with motion sequencing in order to ask whether or not indeed the DLS contains within it neural correlates for syllables, for behavioral grammar, the sequencing, or both. And so together with our collaborators, Bernardo Sabatini and Scott Linderman, who's at Stanford, we have rendered now motion sequencing compatible with all forms of neural recording. So we can basically stick probes or cameras wherever we want in the brain and simultaneously perform motion sequencing. And that allows us to learn about brain behavioral relationships during naturalistic and spontaneous behavior. So I'm just going to show you one example of us doing that. So I'm going to play this movie. This is a mouse that's freely exploring the open field. On the upper left, you see a 3D movie of the mouse. Here we have digitally extracted the image of the mouse from that little arena that I showed you before. And we're going to line the image of the mouse so its nose is always pointed to the left, to the right, and its tail is always pointed to the left. And you can see, this mouse is unusual because it's wearing a hat out of which is coming a cable. And that's because we have implanted in its dorsolateral striatum a miniscope, which allows us to visualize the activity of individual spiny projection neurons within the dorsolateral striatum in real time. You can actually see what this miniscope is seeing inside the brain of this mouse on the right. Each little flash you're seeing represents calcium fluxes that are coming into one of the activated spiny projection neurons in the dorsolateral striatum. And so on a cell-by-cell basis, through this kind of calcium imaging, we can assess ongoing population level activity. And in the center, you see us aligning this neural activity to behavior. On the top are the behavioral syllables as they're being expressed, and we've given each syllable here in this little snippet a color. And on the bottom, you can see traces from individual neurons as they're activated during spontaneous behavior. And I think you can observe one of the key, actually several of the key points related to these data that we've already published here just by looking at this little snippet. So the first is that as animals generate different behavioral syllables, different neural ensembles composed of spiny projection neurons in dorsolateral striatum are systematically dialed in and dialed out. So there's a little pulsive neural activity essentially every time the animal generates a new behavioral syllable. And if we build computational decoders, we can, with a high degree of accuracy at any moment, identify specifically which behavioral syllable the animal is expressing at any given moment in time, based just upon which neurons are active. It turns out that if you do the math, there's effectively a systematic code for three-dimensional behaviors that exists within the dorsolateral striatum. In addition, if we look through the right lens, we can actually see that there's information in the dorsolateral striatum, not just about which syllables expressed at any moment in time, but also about the order in which those syllables occur. And if we lesion the dorsolateral striatum, it actually turns out that animals are still capable of generating all these different 3D motifs of movement, but they're placed entirely in the wrong order. So this centers our thinking about where this structure comes from on the dorsolateral striatum. It suggests the dorsolateral striatum plays this kind of key role in assembling under normal circumstances and actually under sensory-driven circumstances, which I didn't show you, in assembling kind of different behavioral sequences that are built out of these atomic syllables. But that work actually leaves, I'd say, two really critical questions unanswered. So the first question is, why are some syllables actually used more than others? Right? It's clear there's a diversity of usages of these syllables, and we have no explanation for why. The second question is, why do some syllables seem to be quite deterministically connected to other syllables? For example, there's a syllable here on the upper left. I don't have a pointer, but you can see that some of these syllables seem like they're almost obligately interacting in time with other syllables, such that syllable A always precedes syllable B. Whereas there are other syllables, and we would describe those kinds of relationships as being kind of highly predictable or deterministic. So those sequences would be deterministic or predictable. But there are also clearly syllables that are almost willy-nilly connected to other syllables in time, where many, many syllables precede and follow any given syllable. And we describe those sequences as being much more probabilistic, much more random, much less deterministic. So why do some syllables tend to participate in really deterministic sequences? And why do other syllables seem to participate in really random sequences? So that got us thinking about mechanisms that might influence at any moment how often a particular behavior is used. And that in turn got us thinking about the major neuromodulator that influences neural activity in the striatum, which is of course dopamine. And it's well known from the work of many, many labs and groups that dopamine both influences population level activity in the moment in the striatum and plays a key role in unlocking corticostriatal plasticity. And I think it's very reasonable to think about dopamine as influencing the structure of natural behavior, because of course, patients that are denied dopamine, due to Parkinson's disease, don't just have difficulties in initiating new movements, they also have difficulties in sequencing new movements. But the truth is that there's very little known at the basic science level about how dopamine might relate to spontaneous behavior. And what we do know essentially falls into two camps. So there's one set of research that's, I think, highlighted by the work of Dan Dombeck and Rui Costa that suggests that dopamine is somehow involved in triggering new movements. And so if you make a recording of dopamine in the striatum or in the midbrain, the substantia nigra pars compacta, for example, which is the actual structure that houses the dopaminergic neurons that innervate the dorsolateral striatum. If you record from either of these areas, often just before an animal initiates a movement, you see a little flicker of dopamine. And indeed, if you give a strong optogenetic stimulus and induce the release of a bunch of dopamine, that often is sufficient to trigger a new movement. There's another set of ideas that are emphasized by the work of Henry Yin at Duke. These ideas suggest that maybe dopamine is actually continuously fluctuating, and the fluctuation of dopamine in the moment actually encodes the animal's ongoing kinematics of movement. And so in this model, instead of just seeing flickers when animals initiate new movements, you'd expect there to be a kind of continuous variation in dopamine. And that variation should specify some key aspect of the animal's ongoing kinematics. But again, as I've mentioned, there's actually not a whole lot of data in the literature really exploring in a granular way the relationship between dopamine and spontaneous movements. In contrast, we know a ton about dopamine's critical role as a teaching signal. It's clear that the dopamine plays a role in influencing the animal's future behavior in order to allow animals in a goal-oriented setting to maximize their experience of reward and to minimize their experience of punishment. But it's important to note that it's very difficult to think about how dopamine might be playing a role as a teaching signal when we're looking at animals that are just running around in empty buckets with no sensory cues and no tasks and certainly no rewards. And so I think the thinking of the field is really centered on the potential role of dopamine in directly regulating movement during spontaneous behavior. So we ourselves wondered whether we could use motion sequencing to learn something about the relationship between spontaneous dopamine fluctuations in dorsolateral striatum and behavior. And so to do this, we have done an experiment just like I showed you before, where we were recording from the spiny projection neurons that are resident in the dorsolateral striatum. But instead here, we're going to measure dopamine fluctuations in the dorsolateral striatum. And we're going to do that because we're going to use a technique called fiber photometry, which allows us to measure in bulk the dopamine transients that are being experienced by spiny projection neurons in the DLS through the use of the dopamine reporter molecule D-Light1. And so here you're looking at data just like I showed you before. You can see this mouse running around in the bucket. You can see that there's a little fiber coming out of its brain, and that's because we're performing these D-Light recordings. You can think of each of these little flickers you're seeing on the bottom as a phasic transient of dopamine. And we can, again, correlate post hoc these dopamine transients with the animal's ongoing behavior. And I think what you can appreciate just by inspection, right, is that there are way more dopamine transients here than can possibly be explained by movement initiation alone. So there's definitely more going on here. And if we do a bunch of math, we actually have a hard time inferring anything about the animal's ongoing kinematics of movement based upon these dopamine transients alone, with one exception, and that exception is actually the animal's ongoing velocity. And this is a little bit of a complicated plot, so I'll take a moment to explain it. On the y-axis, you're basically looking at the correlation between dopamine and the animal's velocity. And on the x-axis is the time scale at which we're going to bin the data. And what you can see is that if we bin data at short time scales, the left side of this plot, there's actually a negative correlation between dopamine and velocity. In other words, if there's a little burst of dopamine that's kind of focused in a brief moment of time, the animal actually tends to stop. And this is a kind of paradoxical result, but one that has been observed by actually many others in the field, including Josh Dedman's lab at Genelia. But what's interesting is that if we widen the temporal window at which we average these dopamine fluctuations and now look at it not over short time scales, but over longer time scales, we now see the kind of comfortable positive correlation between dopamine levels and velocity that all of us learned about in our undergraduate neuroscience courses. So this is the relationship that we observe in natural behavior between dopamine and just kind of two-dimensional velocity. What about movement initiations specifically? So here on the x-axis is time, where time 0 is the moment at which the animal initiates a new movement after he or she has been still. And the y-axis is just a measurement of these dopamine fluctuations. And what you can see is that indeed, as advertised, as animals initiate new movements, there's this kind of systematic fluctuation in dopamine that accompanies the generation of a new movement. But what's interesting is that we see a much more profound fluctuation in dopamine if we don't just consider movement initiations, but instead consider all movement transitions through the lens of motion sequencing. So now here on the x-axis is time again, but time 0 marks the boundary between any syllable and any other syllable. And you can see there's this systematic fluctuation in dopamine such that as syllables and dopamine levels appear to kind of fall. And then as animals initiate new syllables, there's this kind of burst of dopamine that seems to, if we're going to anthropomorphize, propel the animal into the expression of the next syllable. Now, this pattern is what we see on average. What you're looking at here on the right are actually many millions of syllable transitions that we've averaged in our data set. We actually see something different if we look specifically at the dopamine fluctuations that are observed as animals enter different syllables. So here on the y-axis is a depiction of, on the left, is a depiction of the 40 different behavioral syllables that were identified in our particular experiment. The x-axis again here on the left represents time. Time 0 represents the moment at which the animal enters into a particular syllable. And what you can see is that if we look at syllable-specific dopamine waveforms, that they're actually different. They don't all have the same amplitude or morphology. This top syllable, as you can see here, which is actually a rearing syllable, is accompanied by a kind of rapid rise in dopamine with a very high peak that then kind of slowly relaxes. And this bottom syllable, which I think is a pause, the animal starts out with kind of low levels of dopamine, and then as the pause relaxes, this dopamine slowly rises. And so these are average, these waveforms that I'm depicting here, this plot that I'm showing you here on the left, reflect what we see on average across all of our mice in our experiment. So there's a lot of data averaged into each one of these waveforms. And you might imagine that given that each syllable has associated with it a kind of unique dopamine amplitude and a unique dopamine waveform, that we should be able to use these amplitudes and waveforms that are being observed in dorsolateral striatum at any moment in time to predict at that moment what behavior the animal is generating, which behavioral syllable the animal is expressing. I'll remind you that if we do that exact experiment as I showed you, using activity of dorsolateral striatal neurons themselves, we can make accurate predictions of which syllable the animal is expressing at any given moment in time based upon differences in neural activity that we're observing. And you'd imagine the same thing would be true here. You see these two different shapes. So whenever the animal does the top thing, you'd imagine the animal's rearing. Whenever the animal has a dopamine waveform that's like the bottom one, you'd imagine that the animal's pausing. But it turns out we cannot make these kinds of predictions. And the reason we can't make these predictions is precisely because these waveforms that I'm showing you here are averages. If we look at any individual instance of a behavioral syllable, you see that for every behavioral syllable, there's a huge variety in the dopamine amplitudes and waveforms that are associated with that particular syllable. So if you just look on the upper right here at some of the different waveforms we observe in association with this rearing syllable, you see that indeed often you see these kind of bright positive peaks, but in fact they can be flipped and inverted in many cases. And this pattern is true for every syllable. And I'll mention that this is consistent with my argument that these dopamine waveforms are unlikely to explain the animal's ongoing movement kinematics. That's because if you look at the upper right here, we have 4 different waveforms with very different amplitudes and shapes, but they're all associated with the exact same behavior, the exact same rearing syllable, right? So now we are very confused, right? What's going on here? It's clear that there's a kind of hyper-ultradian rhythm in dopamine where there's these little flickers in dopamine that are accompanying the animal transitioning from one syllable to the next. But it's clear also that these dopamine flickers don't seem to allow us to predict which behavior the animal is expressing at any given moment in time, because there's so much variation on an instance by instance basis in the shapes and amplitudes of these dopamine waveforms. So what might these dopamine waveforms mean? Well, we were worried that actually we'd made a terrible mistake, that we were measuring kind of dopamine fluctuations in dorsolateral striatum, and we were basically making a big deal out of noise. And so we wanted to somehow calibrate our system and look at dopamine fluctuations that are elicited by stimuli that are known to evoke dopamine fluctuations. And so we did something that was very simple. We just took the mouse as it was running around in the bucket, and we threw a bunch of chocolate chips into the arena. And when we did that, of course, the mice will eventually stumble upon one of these chocolate chips and consume this rewarding and surprising treat, which of course is known to be able to shape the animal's future behavior. And we could then measure the dopamine that was associated with the consumption of this pleasurable reward. And we look at the amplitude of food reward evoked dopamine transients in dorsolateral striatum. We see that these reward evoked transients are roughly the same amplitude as the dopamine transients that are observed spontaneously in the absence of a task, or in the absence of rewards, when animals are just running around in the bucket. And that suggests a rather remarkable possibility, right? It suggests that maybe spontaneous dopamine fluctuations aren't influencing behavior in the present by triggering new movements or influencing their kinematics, but instead maybe these spontaneous dopamine transients are influencing the use of transient-associated syllables in the future. In other words, these dopamine transients might be shaping future behavior. If that's true, you would expect that those syllables that on average in a given experiment are most associated with high amplitude dopamine transients, those syllables should be used the most, and those syllables that are kind of at the bottom of that list that are most associated with kind of low levels of dopamine, those syllables should be used on average the least. And that is in fact exactly what we see on the left here. The y-axis is how often in an experiment a syllable is used. The x-axis represents the dopamine transient amplitudes that we observe, and each dot here is a mouse syllable pair. So you're looking actually at all of our data, and you see this kind of lovely positive correlation between how often in an experiment a particular syllable is used and the average amplitude of the dopamine that's associated with that syllable. And critically and crucially, this relationship does not simply hold true on average, but it holds true essentially on a moment-by-moment basis. So as you can see here on the right, imagine you're a syllable like that rearing syllable, and you happen on a given instance of that syllable to be associated with a ton of dopamine. As shown by the orange line, you will use that behavioral syllable more over about the next 3 minutes. If, on the other hand, you express that very same syllable, but you have a low amplitude dopamine peak, you will use that syllable less over about the next 3 minutes. So we know that dopamine doesn't just affect future behavioral choices. We know that if you elevate through the use of drugs, the overall levels of dopamine that an animal is experiencing, that in choice experiments, animals will tend to make more variable choices. And so we wondered whether or not these syllable-associated dopamine transients would somehow influence the choice of which behavior the animal is going to express next. And you might imagine that higher amplitude peaks might prompt the mouse to make a kind of more variable next behavioral choice, or to build a more probabilistic behavioral sequence. And again, that's exactly what we see here on the left. Again, each dot is a mouse syllable pair. On the x-axis is the amount of dopamine that's on average in an experiment associated with that syllable. And on the y-axis is just a mathematical representation of how variable the syllables are the sequences are that syllable participates in, with higher entropy corresponding to greater variability or more probabilism in the sequence. And what you can see is, again, across all of our data, we have this nice positive correlation where those syllables that are associated with the most dopamine are also those syllables that tend to participate in the most variable behavioral sequences. And indeed, this relationship also holds true on a moment-by-moment basis. And so if you're that rearing syllable and you're associated with a high amplitude dopamine transient, you will tend to make a more variable behavioral choice out of the set of behavioral choices you have for your next behavior. And that effect will last a couple seconds, as shown here by the yellow line. And conversely, if you're that same rearing syllable and you're associated with a low amplitude dopamine transient, you'll make a much more predictable or deterministic next behavioral choice. So now I've shown you 3 different correlations between spontaneous fluctuations in dopamine and behavior. I've shown you one between velocity. I've shown you that there's this correlation between syllable-associated dopamine and the use of that syllable, which evolves over minutes. And I've shown you another correlation between syllable-associated dopamine and the variability of behavioral choices, and that evolves over seconds. And we've built both encoding and decoding models that validate these interrelationships. We wanted to do a causal experiment. We wanted to essentially somehow reach in and manipulate at any given moment in time the size of the dopamine transient associated with a particular behavioral syllable. And that would allow us to test the hypothesis that these dopamine-associated, the syllable-associated dopamine fluctuations are actually causal for changing the animal's kinematics, the use of the syllable, or the sequencing that occurred afterwards. To do this, we have recently, through the magic of deep learning, rendered motion sequencing real time. And so now we can just look at animals as they're running around in the open field. And with about a 30 to 50 millisecond delay, we can identify which syllable the animal is expressing at any moment in time. And so we can basically call out what the animal's doing. And that allows us to essentially send a trigger that lets us shine light specifically within the dorsolateral striatum. That in turn allows us to make an optogenetic manipulation that causes dopamine release. And here we achieve that by expressing the optogenetic actuator channel rhodopsin or crimson, depending on the experiment, specifically within substantia nigra pars compacta neurons that project to the dorsolateral striatum. And then we place our stimulation fiber directly over the dorsolateral striatum. And that allows us, in response to light, to cause dopamine release specifically within dorsolateral striatum and nowhere else in the striatum. So it's a very anatomically specific manipulation. In addition, we titrate the amount of light we give to cause dopamine release in order to exactly mimic the amplitude and duration on average that's observed during natural spontaneous behavior. And the consequence of this is that we give a much more subtle optogenetic, we're making a much more subtle optogenetic manipulation than has been done in the past to associate dopamine with behavior. And in our case, we're giving a single 200 millisecond pulse of light at relatively low laser powers. And this is such a subtle optogenetic manipulation relative to what others have done that one might naturally expect that actually it should have no consequence on behavior. And indeed, if we look at the animal's movement kinematics in either open or closed loop, we actually see that these little flickers of exogenous dopamine that we're inducing with light in dorsolateral striatum, these do nothing to the animal's velocity, doesn't make it turn, doesn't make it speed up, doesn't make it rear. From what we can tell, this doesn't change the animal's moment-to-moment kinematics at all, nor does it cause new movement initiations. And this is not because we are terrible at optogenetics, because that's of course a possibility, because if we just leave the light on for longer, mimicking the kinds of things that others have done in the past, now we see the previously observed increase in the animal's speed and the beginnings of the animal turning, and so on and so forth. So this experiment, I think, really argues that these kinds of subtle fluctuations in dopamine that we observe in dorsolateral striatum themselves are not commanding movement initiation or the kinematics of an animal's ongoing movement. However, we see something very different if we ask about how often optogenetic stimulated syllables are used. So here we're going to do this kind of targeted, we're going to target our optogenetic stimulation to a particular behavioral syllable. In this example, we're going to target one of these rearing syllables. And here you can see how often that syllable is used in green. And we're going to do a couple of sessions where we just kind of mock stimulate the animal just to get a sense of how often this particular syllable is used in the baseline. And then we're going to turn the light on. And every time the animal expresses this rear, we're going to give a little puff of dopamine optogenetically. And what you can see is that nearly immediately, the animal recognizes that it's getting this kind of fictive reward, essentially, in response for expressing the syllable. And the animal immediately begins to upregulate the use of that syllable. That holds true in the first session that we do it in. We give the animal a little bit of a break. It holds true in the second session. And we know that this is actually causing the animal to learn something because the latency for the animal to express this targeted syllable in the second session is actually much lower than in the first session. And importantly, when we turn the light off, the animal persists in upregulating the targeted syllable. We've seen this effect now in 8, 9 separate different syllables, so we think it's very general. And I want to just mention kind of how interesting in principle a result this is, because what you have here is an animal running around in a cold, dark, empty bucket with no task or cues provided to the animal at all. And so the only way the animal has of knowing what was associated with the little dopamine we gave it is basically proprioception, right? The animal can only know what it's done through its understanding of its own body, and yet that's sufficient for the animal to recognize what it's doing and to enable upregulation of the specific behavioral syllable that we've targeted. And I want to mention that we've done a bajillion controls to show that the stimulation that we give affects only the targeted syllable. And it turns out it doesn't affect even related syllables morphologically. And so it's clear that the DLS-associated dopamine transients are sufficient to upregulate the use of an associated behavioral syllable. And it also, from this experiment, tells us that these syllable-associated dopamine transients are also sufficient to induce an increase in the variability of subsequent behavioral choices. So it turns out that the way we do this experiment is we only give dopamine on about half the instances of any given targeted behavioral syllable. And so we have kind of optogenetic stimulus in the green shown here, but we also have catch trials where we don't actually give dopamine to that same syllable. And you can see here, as was observed in the endogenous data, that indeed dopamine transiently increases sequence variability. So I've now showed you kind of causal evidence. The dorsolateral striatal dopamine probably doesn't influence kinematics, but instead likely influences much more structural aspects of an animal's behavior, including sequence variability as shown here, as well as how often a syllable is used. And that suggests that perhaps these ongoing fluctuations in dopamine that we observe in dorsolateral striatum are actually structuring the animal's ongoing behavior. And we wanted to test that formally. So in collaboration with Uchida, we have built a reinforcement learning model where a reinforcement learning agent based upon actual dopamine syllable associations that are observed in some training data learns to make behavioral choices. We train this reinforcement learning agent through simple Q learning rules. We initialize its Q table, which is equivalent to the transition matrix that governs how any given syllable will transition into any other syllable uniformly. That's the big red block you're seeing here in the middle. You'll notice that we're only going to consider in this analysis the top 10 used syllables because we're a little bit data limited. And there's some blocks that are not considered that are in black. And that's because those are behavioral transitions that normal mice just never make. And so we can take a reinforcement learning agent, feed it information about syllables and dopamine transients, and mathematically treat these dopamine transients as if they're rewards and therefore in the future should increase the use of that particular behavioral transition, effectively that syllable, and increase the variability of future behavioral choices. And if we go through this training procedure using Q learning rules, you can see that this fictive reinforcement learning agent actually learns a transition matrix that effectively governs the behavioral choices this animal would make in the future if this agent were like an animal, a real animal. And then we can compare this transition matrix, which is built based on the premise that dopamine reinforces syllables and increases sequence variability. We can compare this to actual behavior that real mice generate. And what we see, to I think our own shock, is that there's an incredible correlation between the transition matrix that's learned by the synthetic reinforcement learning agent and the behavioral choices that actual real life mice make. And that suggests this kind of possibility that indeed mice are interpreting these fluctuations in dopamine as a kind of teaching signal that helps them build the architecture of their behavior as it flows over time. I want to mention one of the clinically relevant things that actually pops out of this analysis. So the training procedure here includes a role for dopamine both in reinforcement and in inducing sequence variability. those two things are seemingly contradictory, right? Like if you increase the use of a syllable, behavior should be more predictable, but also you get this little increase in variability that it just occurs over seconds, but that increase in variability seems to kind of contradict the fact that dopamine's association with increases in usage should make behavior more stable, not more variable. And so we built reinforcement learning models in which we didn't allow dopamine to induce variability. We just allowed it to be reinforcing. If you do that, mice effectively develop habits. They perseverate unnaturally on just the first syllable that is associated with a high amplitude dopamine transient. And so it appears that like the ability of dopamine to both cause reinforcement and to induce variability allows animals to break out of what would otherwise be repeatedly used behaviors and to explore the behavioral space. And one might imagine then that this effect that dopamine seems to have in increasing transiently behavioral variability might be defective in scenarios in which animals or humans repetitively express behaviors. And that's a hypothesis we're eager to test. For reasons of time, I'm actually going to skip over this part of the talk and just go to the end, so we have some time for questions. I have shown you here that we can build this technology called motion sequencing that decomposes behavior into its constituent elements. That reveals its overall global structure and shows us that the brain really is imposing structure on behavior. I've shown you that the dorsolateral striatum is a key node in kind of creating and assembling the structure. And I've also showed you a provocative unpublished data that suggests that ongoing fluctuations in dopamine in the dorsolateral striatum actually impose structure on high dimensional spontaneous behavior, even in the absence of a task or experimental afforded cues or any forms of reward or punishment. In other words, the very same circuits and the same kinds of computations that support the ability of a human or an animal to make appropriate decisions in a low dimensional decision-making task that yields reward, those very same circuits and computations allow animals to build high dimensional spontaneous behaviors in the absence of a task or in the absence of a reward. And for us, there's really two key questions. The first is, of course, what is governing these fluctuations in dopamine and thereby imposing structure on behavior? I'm happy to discuss that in the question and answer period. The other thing I'll mention that I skipped over, but I'll just mention the result, which is that actually individual animals turn out to vary in terms of their sensitivity to dopamine. And that actually is responsible for quite a bit of the individual variation we observe in animals' behavior as they explore the open field. And one question then is, why does dopamine sensitivity vary? And of course, understanding why dopamine varies during natural behavior and why the sensitivity of dopamine might vary should go a long way to teaching us about how spontaneous behavior is built. Just to end, I'll mention that this work was done by three amazing people in my lab: Jeff Markowitz, who's now a faculty member at Emory and Georgia Tech, and two graduate students, Wynne Gillis and Maya Jay. All the work I told you about today was a collaboration between my lab, Bernardo Sabatini's lab at Harvard, now Uchita's lab at Harvard, and Scott Linderman's lab at Stanford. This is the rest of the lab, and I would be delighted to take questions.
Awesome. Thank you so much. Really fascinating presentation, as always. Maybe I just, out of self-interest, and I wonder if you wouldn't mind just taking a couple minutes to show us that variability result that you mentioned towards the end. Moderator's discretion on this, but I think that's really fascinating. The idea that, because we've been hearing throughout this conference too, of really emphasizing that these methods, both in humans and animals, allow you to get at these questions. Why do individuals, how do we use these to kind of explore heterogeneity at the individual level? So feel free to maybe just comment, or if you want to share yours.
I will do that. I'll share my screen. I will just start with this and just show you 2 slides worth of stuff. Okay, so typically in systems neurobiology experiments, you train an animal in a task, and then when the animal reaches some behavioral threshold criterion, you consider that animal well-trained, and then you study what's going on in the brain, right? And so, kind of by convention, we throw out animals that for whatever reason cannot learn or learn poorly, because they kind of mess up our analysis, and we do that in part because we just assume there's individual variation in learning, And we, in part, attribute that to our own failures, right? To variability in the housing of the animal, the way we train the animal, and so we just kind of ignore the fact that animals have variability in their learning. But when we submitted this paper for review, we described all of the data, and you can see here on the left, and just replotted in a slightly different way on the right, that in fact there's quite a bit of variability in terms of, on a mouse by mouse level, that's what you're looking at in each one of these green dots, in how effective optogenetic dopamine is at causing that syllable reinforcement effect that I told you about. Okay, so mice seem to vary. Individual mice seem to vary in terms of how effective optogenetic dopamine actually is at promoting learning. And you would assume that this is because maybe our fibers in the wrong place, or there's other reasons for variability. But we hypothesized, prompted by the reviewer, that perhaps there was something deeper and more interesting going on. Perhaps what was differing amongst these mice is actually their intrinsic susceptibility to dopamine. And I'll remind you that at the beginning of my talk, we could show that dopamine transients cause the use of associated behavioral syllables for about the next 3 minutes. Now that result is on average across all of our mice, right? It turns out that if you do that same calculation for individual mice, there's actually a great deal of variability in terms of how strongly a syllable-associated dopamine transient will influence future behavior on a mouse-by-mouse basis. In fact, if you look here in the upper left where it says counts, that's really how often a syllable is used. And that curve you're looking at is actually a curve describing the variability across individual mice and how much endogenous dopamine fluctuations change future behaviors. In some mice, it's really a profound effect. Instead of dopamine transients influencing usage over 3 minutes, it's actually 10 minutes. In other mice, dopamine doesn't seem to affect behavior as much. Maybe it reinforces the use of that syllable over the next minute. The effect is always positive. We always see a reinforcement effect, but you can see this kind of distribution on the upper left, describing individual variability and how strongly endogenous dopamine seems to influence syllable usage. I'll remind you also that syllable-associated dopamine causes increases in sequence variability. You can see on the upper right here that there's a variation in how strong that effect is across individual mice. Again, some mice are really affected by dopamine. They make very variable next choices when they experience a dopamine transient. Other mice are more predictable, and actually dopamine has less of an effect on them. On the lower left, you can see us correlating how strongly the effect of dopamine is on syllable usage, which again evolves over minutes, and how strongly dopamine affects sequence variability, which again evolves over seconds. Each blue dot here is an individual mouse, and you see this near perfect correlation. Mice that are affected by dopamine in terms of their usage are also those that are affected by dopamine in terms of sequencing, and vice versa. And that kind of suggests there's like a single latent factor that determines how sensitive a mouse is to dopamine. And if we ask, whether we can use these endogenous measures of dopamine sensitivity to predict learning, in fact, we can. So those mice that are most sensitive endogenously to fluctuations in dopamine are precisely those mice that do a good job of learning when we add a little bit of extra dopamine. And actually, we can kind of model these data out and show that it looks like the way the brain is interpreting endogenous and exogenous dopamine is just by addition. The effects that we're seeing behaviorally are really the result of optogenetic dopamine being added to endogenous dopamine fluctuations. And this is incredibly exciting because it suggests that there's this kind of latent variability in sensitivity to dopamine, even in mice that are genetically identical and housed under the same conditions. And in this case, they're all males. And so they're the same sex. And so we're really interested in trying to track down epigenetically or through other mechanisms, what's accounting for this variability and sensitivity. And it also suggests another interesting possibility, which is since endogenous dopamine sensitivity actually influences future behavior, we should be able to look at mice by motion sequencing and make predictions about how good they are at learning. And that's pretty amazing, right? That we in principle could look at the open field behavior of an animal and make an accurate prediction about its future ability to acquire a task. And that's something that we're deeply interested in looking at directly.
Very cool. Maybe I could just follow up on that one. So, you're making it sound like this is an animal specific thing, but I can imagine that like the time course between when you do the initial, dopamine spritzing and then the actual learning task, like that's probably relatively near in time. I guess I'm just curious, were you able to look within an individual animal over multiple days of assessment? Is this a stable trait-like thing, or is it the case that like if you sleep deprive the animal, some of this goes away, or if you make the, one of the questions in the chat is whether the emotional state of the animal affects this at all. So I guess, maybe you could just comment a little bit more on this. Do we have ways, or have you explored ways of seeing whether that sensitivity is experimentally shiftable? Can you induce anxiety or cause, more of a depressive phenotype?
That's a fabulous question. So I'll answer that directly. So like, that's exactly what we're interested in looking at. I will say there's like already one hypothesis that we have. We haven't done any experiments, but you can imagine it's a real hypothesis. So, just remember what we're doing here, right? Like we're implanting a mouse with like a cable to manipulate dopamine, and we're separating the mouse out so that, because otherwise they'll eat each other's cables, and we make them live alone for a while. That's highly stressful for mice. And so you might imagine that like this latent factor is revealed really through kind of individual variation in stress responses. Some animals may just be able to handle the stress of the whole procedure better, and that lets them be better learners versus not. And that's one of the kind of, that's definitely one of the kinds of hypotheses we're interested in testing. What I'm reporting to you here are the results of actually a month's worth or 6 weeks worth of experiments on the animals. So it's probably a stable trait within an animal. And so there's many possibilities, right? It could just be epigenetic variation that occurs during development. It could be stress related. It could be about kind of long-term emotional variability. We're interested in all those possibilities.
Cool. And within a month still would presumably be, I assume these animals are roughly at the same developmental stage. So do you think
they're all
Yeah, I guess that's the question. Is this something, maybe it's a speculation, but do you think this is something that's just systematically shifted as a function of like transition to adulthood? Because you can imagine that like at certain stages in development, you want to emphasize the reinforcement quality of the dopamine versus in other stages, it's, dopamine might enhance variability to kind of increase risk-taking or foraging behaviors or something like that. But
so Justin, you're gonna love what I'm gonna say next. So we have done MoSeq life to death for mice. And so we have like this very large scale, basically petabyte scale atlas of the evolution of mouse behavior. And we're writing it up now. One of the things we observe is, of course, as animals enter into old age, their behavior crystallizes. There's structural changes in individual variability. And what accompanies that is a change in their sensitivity to dopamine. So when we look at the relationship between dopamine and variability, that actually is altered and attenuated. And it looks like the relationship is such that like animals might be making more predictable behavioral choices because that relationship has been altered systematically, which I think is a kind of a very exciting way of thinking about how things might actually change over time. And it might explain the fact that as I get older, I'm just much more less behaviorally flexible.
Sure, we all, get entrenched into habits, but then there's a certain amount of, needing to stay somewhat agile that I guess confers some health benefits too. But well, this is great. I guess one other question that is in the Q&A, I guess, not sure exactly the question, but it says, could these approaches help quantify cognitive reserve? Maybe this kind of gets at the question of, whether if you can look at individuals who maybe can learn more readily, that prevent them from being susceptible to maybe more stressors and things like that.
Yeah, so I'll just pop up one level here. I think one of the exciting implications of this work, which I hope isn't lost on people, is that for the history of behavioral neuroscience, we've treated open field behavior like it's just locomotion, like it's a univariate thing that you just use to measure whether the animal can move its limbs around. And maybe we can like impose some human based biases and interpret it as if open field entries reflect anxiety in some way, for example, right? That's the thinking traditionally around the use of this assay. In truth, the fact that dopamine dynamics are specifying behavioral dynamics over short and long timescales, and that really tells you that there's certainly a component of short-term memory that's coming to play here in terms of the animal's actual open field behavior. And I think that begins to build the argument that this behavior that we thought was very simple, the animal running around in the bucket, actually is a cognitive task for the animal. The animal's exploring the bucket, probably building an internal map of the bucket, and the animal's own movements reflect the creation of that map. And so it's a much more, through the right lens, even this very simple experiment, this very reduced experiment, reveals something really fundamental about the cognitive status of the animal. And so we're very interested in exactly this question, like, can we begin to think about open field behavior as telling us something fundamental about the animal's overall cognitive status. We think that should be true given what I've told you, and we're continuing to work along those lines. So I think it's a great question.
Well, it's fascinating. It reminds me, think about kids development too. It's what happens on the recess, in the open field where you actually see a lot of the kids, social emotional learning, coming to fruition.
Yep.
I'm really glad this close to Halloween that you also said that you literally are throwing chocolate chips into the field. So as I see my kid, trick or treating this week, and I'm sure I'll be thinking about this or ways we can study humans in their own kind of version of this. But amazing. Well, thanks, Bob. I guess really appreciate your coming back to TIPS and sharing this exciting work, which I know is going to be coming out in a publication fairly soon. So we'll keep our eyes out for it. So
Thank you. Thank you so much for having me.
absolutely.